Hela Azaiez is a senior scientist at the Molecular Otolaryngology and Renal Research Labs at the University of Iowa in the United States of America, where she is currently leading the hearing research and diagnostics teams. She received her master's in Biological Engineering and Ph.D. in Human Molecular Genetics at the University of Sfax in Tunisia, conjointly with the University of Iowa. So Hela is going to talk to us today, unraveling the genetic complexities of hearing loss to empower precision medicine. We welcome you, Hela, and the floor is yours.
Thank you, Mariam, for that nice introduction. Just a second, moving my thing here. Voila. Oops. Do you see my slides?
Yes, we can see it here.
Okay, thank you. So I would like to start by thanking the scientific and the organizing committees for offering me the chance to be part of this incredible conference. It's a real pleasure to be able to present and share with you some of the work we have done exploring the genetics of hearing loss and its complexities, and also present some of the challenges we have encountered and the tools and strategies we have developed to overcome them in order to fill the promise of precision medicine. Since the launch of the Precision Medicine Initiative in 2015, genetics and genomics were propelled to the forefront of disease prevention and treatment efforts. Precision medicine, as you know, represents a shift from the standardized one-size-fits-all medicine to a personalized approach to healthcare, in which medical decisions, interventions, and treatments are tailored to individuals based on their genomic data, environment, and lifestyles. For hearing loss rehabilitation therapies, and we are talking here about hearing aids and cochlear implants, are not suitable for all individuals and do not work with the same efficacies for all. So hearing loss is the most common sensory defect in humans. It affects 430 million people worldwide, and by 2050, that number is predicted to reach 2.5 billion. This is according to the latest World Report on Hearing 2021, a couple of months ago. And we could see that all continents are affected. As you see here, this is the region here in, I guess it's yellow. This is the East Mediterranean region and North African region. So these are in here is the Arab countries. And we could see that the number of people suffering from hearing loss is going to double by 2050. So hearing loss, the incidence is 1 in 1,000 births. And both environmental and genetic causes have been involved in the etiology of hearing loss. The portion with which genetic causes are responsible for hearing loss depends on countries. In high-income countries, the genetic causes account for 80%. In low-income countries, genetic causes account for only 50%, and the rest is going to be environmental causes such as congenital rubella, CMV, ototoxicity, and others. Of the genetic causes of hearing loss, 70% are non-syndromic. It means they are only the only system that is affected is the auditory system. In 30% of these cases are other system and other organs are affected, such as Usher syndrome, and we will talk a little more about this one later. Usher syndrome is characterized by severe to profound congenital hearing loss, and retinitis pigmentosa at puberty, and also vestibular dysfunction. Some other syndromes are also rare, like Waardenburg syndrome, and this is here like with the white forelock characteristic and bright blue eyes, and Pendred syndrome. Of the non-syndromic hearing loss, autosomal recessive forms represent 80%, and dominant forms 20%. One of the striking characteristics of hearing loss is that it is clinically heterogeneous. So it could be, as we just talked about, it non-syndromic or syndromic, and here are certain syndromes where hearing loss is one component. It could be sensorineural or conductive. It could be the onset of hearing loss could be different from one person to the other. The severity is different. The frequencies affected are different. And here we have an audiogram. Here is the hearing threshold in decibels, and here are the frequencies. And you could appreciate here, this is what we call the audio profiles. And each, they are different ones. They are very, very different. We could have low frequency hearing loss, whereas the high frequency, it's normal. We could have the opposite. Lower frequency, there is normal hearing, and then at higher frequency, there is hearing loss. So it differs from one individual to another, and it also depends on the genes involved. The progression is also different. Some have congenital stable hearing loss. Others, hearing loss will develop later in life and is progressive. It's not surprising then that this complex phenotypic landscape is mirrored on the molecular level as well. Over the last 2 decades of genetic research into hearing loss, a lot of genes have been identified. And the rate of gene discovery has accelerated since the advent of next generation sequencing, with over 150 genes identified to date. And of those 120 genes for non-syndromic hearing loss alone, over 700 genes for syndromic hearing loss, and in those genes, over 16,000 reported pathogenic and likely pathogenic mutations. These genes that we have shown in the last slide encode for a wide group of proteins with diverse functions, expressions, and structural function. If we see here, for example, this is the ear, this is the cochlear, this is the organ responsible of hearing. A section here will show us the organ of Corti and the different cells involved in hearing. We have the outer hair cells here in blue, the inner hair cells. We have the supporting cells, and here is a zoom in of a hearing cell. And as I said earlier, different molecules have very specific expression and specific cell type at specific timing with specific functions. To add another layer of complexity is hearing loss is really genetically complex. So one gene could cause different phenotype. For example, MYO7A gene, mutations in MYO7A gene could cause Usher syndrome type 1. As I said earlier, this is a very severe phenotype with severe and profound hearing loss, retinitis pigmentosa, and vestibular dysfunction. However, other mutations in MYO7A also cause non-syndromic hearing loss, autosomal recessive, DFNB2, and autosomal dominant 2. To add more complexity to that, there are all the genes that also cause Usher syndrome type 1. This is USH1C, USH1G, cadherin 23, protocadherin 15. And then we keep building on that complexities. These genes by themselves, all the mutations in them also are responsible for autosomal recessive non-syndromic forms of hearing loss. The inheritance of hearing loss is also complex. We have monogenic forms, as we talked about earlier, but we have also the complex and multifactorial forms. These are age-related hearing loss, nose-induced hearing loss. The prevalence of hearing loss, depending on the type and depending on the syndrome and depending on the gene, could go from really ultra-rare to common. And for common, we are talking about here age-related hearing loss and noise-induced hearing loss. So the promise of precision medicine in the auditory field is immense because of this extraordinary phenotypic, genetic, and mutational landscape. And understanding the intricate relationship between these mutation, gene, and phenotype was one of the aim of our work. And as we said here, just a summary. So when we talk gene, this is genomic landscape, different diverse function for the genes, diverse mechanisms. The mutation, they have different mutation type and effect. Phenotypic spectrum, syndromic versus non-syndromic, dominant versus recessive, onset, severity, progression. And we are trying to understand these intricate relationships. We will start first by understanding the clinical validity. And this is really important. So for hearing loss, we have a website, Hereditary Hearing Loss, that actually reports all the genes that have been identified to date and involved in hearing loss. However, as you may all know, there are a lot of publications almost every day about novel genes for different diseases. The issue here, not all of them provide convincing or strong evidence for the validity of this relationship between the gene and the phenotype. So for hearing loss, we created the ClinGen hearing expert panel. And this, it encompasses experts from around the world, all of them expert on the genetics of hearing loss. And we meet very often. We started by meeting almost every week, and now it's once a month. And what we do is we study and we investigate all the published literature that has been out to look for and literally score the evidence provided to link a specific gene to specific phenotype. Not surprisingly, our data has been published recently, and we showed that actually only 15% of the genes that have been published have a definitive evidence of their association with the hearing loss. And if we add the ones with the strong evidence, so 57% of the genes that are out there for hearing loss have a strong enough evidence to link them to hearing loss. All the rest have a moderate evidence, and 26 and 2%, so this is 28% have limited, disputed, or literally just refuted. Their role in hearing loss was refuted. This is very important because when you have a patient in the clinic and you are trying to figure out what's their hearing loss, you want to order a panel or whether exomes or genomes, but you want to actually provide data that is robust. You cannot report variants in genes where the evidence is limited or disputed or refuted. So knowing actually what genes need to be reported back to patients is really important. In our lab, and over the past decade, we have developed and refined a custom genetic pipeline. So we have created a specific panel for hearing loss, and we use it, as I talked earlier, about those genes with the strong and definitive evidence to their association with hearing loss. We have screened over the last decade over 7,000 samples of individuals with hearing loss, and we follow the next generation sequencing using a panel having 225 genes. This is including all non-syndromic hearing loss genes and some of the most common syndromic forms. After bioinformatic analysis and Bayesian filtering, we get to the really most important part here, which is variant classification. And for this, we meet this multidisciplinary team meeting with a scientist, a geneticist, bioinformatician, and genetic counselors to discuss the results and the genetic finding for each patient, and then to generate a report. As you all appreciate here, is the genetic variant classification is crucial to accurate genetic diagnosis, and it really represents a major challenge. So here, depending on the panel you are using or the pipeline you are using, you might end up with from 500 variants to 4.5 million variants if you are looking at the genome. And over the last decade, the sequencing technology is no longer the limiting factor in providing genetic data. It is actually the interpretation and report generation for all of these variants. And when I talk about variant classification, we follow, or we talk here about the American College of Medical Genetics that came up with a 5-tier classification of variants, and that is so variants are classified into 5 categories: benign, likely benign, VUSS, likely pathogenic, and pathogenic, to indicate their likelihood to be associated with the disease. And again, here, when these came up at the beginning, they were non-specific, so they were not specifically for hearing loss. For this, we have actually also created a hearing loss variant curation panel to help set specific guidelines that are tailored specifically to hearing loss. So doing that, as I said, there are over millions of variants in the genes involved in hearing loss. So we needed somehow a tool that will automate the classification and help with the classification of variants. So we come up with the deafness variation database. And in that, it's a bioinformatic pipeline. And for this, we include all the deafness genes. We run them through a BEP pipeline to assign clinical significance, diseases, minor frequency, conservation, deleterious scores. We get data from ClinVar, from AGMD. And then we also manually curate those variants. And then we put them in a publicly available database. That's, again, the Deafness Variation Database, or DVD. So currently, the DVD have 2.5 million variants in hearing loss associated genes. Of those, there are 16,000 pathogenic, 4,000 likely pathogenic. All the rest is VUSS and likely benign and benign. And we have over 3,000 user years. And it's really helpful for both clinicians and patients. So doing so, we actually were able to reclassify over 1,200 variants, and over 300 variants were medically significant, which means they changed category, either going from likely pathogenic to benign, or the other way around, from benign likely benign to pathogenic, or from VUS to pathogenic likely pathogenic. Importantly, some of these reclassification that are medically significant affected the genes that are really involved in Usher syndrome. So this is really important because this is, again, it's a very severe phenotype. So next, after we did that, we constructed a database that has all variants in hearing loss. We sought actually to define the mutational signature of each one of these genes. So of those almost 2 billion, actually, 4 million variants, when we looked at their minor allele frequency, surprisingly, 96% of coding variants in these hearing loss genes are actually, they have a minor allele frequency less than 0.5%. That means they are rare. So we cannot really use minor allele frequency. It's not going to be that helpful, or we cannot use solely based on minor allele frequency to say, to be able later to classify a variant. An important thing is most variants are missense. So 62% of all variants in hearing loss genes are missense. That creates actually a very interesting challenge, because thus we couldn't classify most of these variants. So almost 3/4 of all variants in hearing loss genes remain classified as a VUS. And the VUS is again, it's a very challenging thing. Here is a summary of the data. So of all the variants that are less than 0.5%, most of the variants here in orange are VUSs. So we kind of took a step back and tried to look at the mutational spectrum of these genes, specifically each gene. So here we have the 40 most prevalent genes in hearing loss, and here is all the variants classified whether by type, whether they are loss of function, missense, or synonymous. And when you look at them, it's kind of similar. It's a uniform distribution of loss of function, missense, and synonymous. However, this picture completely changes when we only look at pathogenic and likely pathogenic variants. And here a different picture emerges, where different genes have different contribution, like the type of pathogenic or likely pathogenic variant types is different from one gene to the other. For example, if we look at ICTG1, all the mutations, pathogenic variants that have been reported are missense variants. If we look at DFNA5, all the pathogenic mutations are actually synonymous or splice sites, so it's affecting the splicing. If we look at other genes, for example, T-SPEAR, all the mutations are actually frame shift and loss of function mutations. So this is really very helpful, because before that, no one did that. And this is really helpful. When we find a variant in a specific gene in a specific patient, we go back and look, what is the mechanism of action of this gene? What are the mechanism of action of different mutations in these different genes? So having done that, Now, as I said, we need to make a clinical correlation between the mutation we found in a patient and correlated to their phenotype. For this, as you remember, and I mentioned earlier that hearing loss, there is like a great phenotypic clinical heterogeneity associated with hearing loss. So the next challenge becomes, how do we correlate the genome to the phenome? How do we check the concordance between the type of hearing loss exhibited by a person and the genetic variant identified through genetic testing? To address this challenge, we developed AudioGene. AudioGene is it's a machine learning algorithm that uses audiometric data to predict genotypes. For example, here, you could see here, this is a 3D audiogram, and this is again using audio profile surface. What we have done is actually, it was 2-dimension before with one just hearing loss and frequency. We added the 3rd dimension, which is the age, and now we have this. It's a surface now. What makes it really interesting is we can literally plot a patient's hearing loss, that this is this red line here, and compare it to what is known for that specific gene. So what this it's actually what this helps to do, it allows the prognostication over time. So we can tell them at this age you has that much hearing loss. When you are getting older, your hearing loss is going to deteriorate at this pace, and by this age you will have this specific hearing loss. What AudioGene is also great for is it's essential for clinical trials. Why? Because we could use it actually to make a very accurate picture of the natural history of the specific gene and the specific phenotype. So having done using the methodology I have described of all the tools we have developed, and after analyzing over 5,000 samples to date with individuals with hearing loss, we come up with diagnostic rate. And we showed here the total diagnostic rate is around 43% for hearing loss. The total genetic diagnostic rate is 43%. And that depends on the inheritance, with autosomal recessive having a higher diagnostic rate than autosomal dominant. And what is also striking is that the diagnosis rate is different by ethnicity and ancestry, with African Americans and Africans having the lowest solve rate. You could see here that populations and that have a higher rate of consanguinity, this is for people with Arab ancestry, Turkish or Iranian, have actually the higher diagnostic rate. Now, if we look at the genetic etiology of hearing loss worldwide, this is a general hearing loss. GJB2 is the gene that accounts for most cases of hearing loss worldwide. That has been already shown before. For some population, it goes all the way to 50%, which means half of the cases of hearing loss are accounted for by mutation in GJB2. The second one is stereocylin and etc. We also showed that these 6 genes account for 50% of all cases. But we have also shown that 84% genes account for only 10% of all cases, and these represent rare causes of hearing loss. Some of these genes have only been reported once in one family or just in one country. Now, when we look at the genetic etiology by ethnicity, a different picture emerges. And you could appreciate here how different it is, the contribution of each gene for these different ethnicities. We have the Iranian, Hispanic, Nigerian, American, and you could see that it is different. I'm going to dive a little more into the genetic etiology of hearing loss in Arab countries. As we see here I have not included GJB2 just because for all of them, it is the most prevalent. And sometimes there is bias in ascertainment because it's the first gene that has been identified and it has only one exon. So historically, it has been screened first. So most people will get screened for GJB2 first, and when it is negative, they send it for comprehensive genetic testing. So that's kind of the data was biased. So for this, for these calculations here, I removed GJB2, but I will talk about it later. So if we look more specifically at the Arab things, so here is the thing. So Arab populations contributed to the discovery of 25% of novel deafness genes. That's fantastic. That's great. However, since then, I mean, the published literature, and there is relatively insufficient numbers of genetic studies, which makes Arab ancestry extremely underrepresented in genomic databases, which in turn leads to inaccuracies in variant interpretation. If we go back here to the hearing loss, so here we have the MORL, our cohort, and we see that there is different. The only comprehensive analysis was done in Palestinian cohort, and it shows a completely different picture than the literature review ones. And again, this is because of bias. Another way actually to look into to see what is the etiology of hearing loss in Arab countries, to look at the carrier rate for pathogenic, likely pathogenic variants in NOMAD. And you could see here in red, this is the Middle Eastern, the Arab countries. And we can actually, looking at this, kind of guess what are the most prevalent genes and mutation genes in the Arab population. Another thing is, when we look at universal newborn hearing screening, it has become the cornerstone for early diagnosis of hearing loss. It permits like timely intervention, and it's quick, inexpensive, and non-invasive. However, when we look at the country, it has been only implemented in 55 countries, and that's not enough. Some limitations still have for newborn hearing screening, that it might miss mild to moderate hearing loss, and that it does not provide etiologic diagnosis or prognosis. And most important, it also misses non-syndromic deafness mimics. As I said earlier, Usher, since the retinitis pigmentosa develops later, we will completely miss that it is Usher. We would think it is just non-syndromic hearing loss. So the current, and what we are pushing now for, is the concurrent newborn hearing screen and genetic testing at the same time. Not because before it was, if kids fail or newborns fail physiological screening, we will then do genetic testing. We propose to do them concurrently, again, because both of them can be done quick and cheap. And one important thing also about genetic testing is that it could really be the base for gene therapies. Most gene therapies are adapted therapies. This means they are made for a specific mutation or specific genes. Here are the mutation-specific approaches like the antisense oligonucleotide, gene editing strategies, and then they need to be adapted. What is the gene, what's the mutation, and what is the mechanism? Currently, there are several successful gene therapy in animal models. 3 of them actually are going through clinical trials. The Myo7A was discontinued. The study was stopped because of some problems with efficacy and safety. But right now there is a clinical trial for H2A and another one for O2F. And again, that's why it's really important to have everyone screened so they have a chance to actually to be part of clinical trials and for gene therapy later on. My last thing, and I should be done fast. There are still some challenges that remain. As I said earlier, diagnostic rate is about 45%. So what is happening to all the 50 other 5%? So that could be different reason why we cannot reach the 100%. One of them is the technology, the capture bias, bioinformatics, the read lengths. One is the phenotype. As I talked about, there is a huge pleiotropy. There is gene-disease relationship that are different. There could be novel genes yet to be identified. We don't know them. The effect or the role of epigenetics, the hidden heritability. And I don't have time to talk about any of that or the work that we have done there. I'm just talking here about, for example, novel transcript and exons. We have done single cell RNA-seq on inner hair cells and outer hair cells. These are specific to the cochlear. And we show that actually we found 2 exons that have not been reported before. And now work is ongoing. We have created the mouse model, and work is ongoing to look for mutation in these patients. The role of genetic modifiers, here again, I cannot go into that much details, but we have seen in humans that there are different progression and severity in some patients with TEC-TE mutations. We created the mouse models, and we have actually identified the modifier for the phenotype for TEC-TE. The structural variants, we actually have shown, we included CNV analysis in our pipeline, and we have shown that actually CNVs, or copy number variation, account for 1 in 5 diagnoses of hearing loss. Finally, just want to some messages take home is the genomic and mutation landscapes of deafness are diverse and complex. The ability to provide precision medicine in routine clinical setting depends on the availability and affordability of genetic testing, capacity building to address the gap and disparity in human educational technological resources worldwide, accurate interpretation of variants, shared knowledge base, and the ability to actually translate this to patient care. For this, I will stop here. I would like to thank everyone at the Molecular Otolaryngology and Renal Research Laboratories and all our collaborators from all over the world. Thank you.